I'm Dr. Michael DiGiovanna at the Yale School of Medicine, and I'm affiliated with the Yale Cancer Center and the Smilow Cancer Hospital. And I've been funded to do breast cancer research by the Connecticut Breast Health Initiative for the past 3 years. And I think of my own research as dating back in time to the 1890s, when a physician named Dr. George Thomas Beatson took care of a young woman with very advanced stages of breast cancer that had metastasized throughout her body. And Dr. Beatson removed her ovaries, and that caused her cancer to regress. We didn't understand till decades later why that happened, but the explanation was that removing the ovaries removed the primary source of estrogen in the body, and it turns out that many breast cancers are driven by the hormone estrogen. Around the 1970s, we actually developed drugs that could do the same thing. These are drugs called anti-estrogens, such as tamoxifen, and that was a treatment that has been used ever since that time for treating women with the type of breast cancer that's driven by estrogen. That's what we call estrogen receptor positive tumors. Around the 1990s, another receptor was developed that was important in breast cancer, and that receptor was known as HER2. And it was discovered that about 20% of breast tumors make too much of HER2, and that drives the cancer to grow very quickly and aggressively. And in the late 1990s, a drug was developed that could neutralize HER2, and that drug was called Herceptin. And Herceptin has been a major breakthrough in improving the cure rate for HER2-positive breast cancer. And it turns out that with conventional chemotherapy treatments, which were developed in the 1940s initially, that kind of treatment didn't start curing people until doctors began combining different drugs together. And that was in the 1960s, when conventional chemotherapy could begin to cure people with cancer. And so my thought, and the thought of many people in the breast cancer field nowadays, is that combining these newer types of therapies and combinations might be the next real step forward in the next breakthrough. These medications like Herceptin and tamoxifen we call targeted therapies for cancer. They're completely different from old-fashioned chemotherapy. And there are at least a dozen of these targeted therapies that are now in use for treating different types of cancer, and many dozens more that are in the development pipeline. So one natural idea was to combine drugs like tamoxifen, anti-estrogens, with drugs like Herceptin. And in the laboratory, that combination was very synergistic. But in treating patients with breast cancer, so far the clinical results haven't been as dramatic as we were hoping. So that led me to wonder what other targets might exist that we might be able to develop new drugs for to use in combinations like that. And it turns out that there is one other type of receptor called the insulin-like growth factor receptor, or we abbreviate it IGF-1 receptor. And it turns out that this particular receptor can be present in breast cancer and can interfere with how well that Herceptin works or tamoxifen works. So this became an ideal target to combine with the other types of drugs. So in my work, previous to joining up with the forces of the Connecticut BHI, I had shown that combining new types of drugs that block that IGF-1 receptor, when combined with anti-estrogen or combined with anti-HER2 drugs, was synergistic in the ability to kill cancer cells in the test tube. When we combined tamoxifen and Herceptin, it stopped the cancer cells from growing, but it didn't kill them. But if we also blocked the insulin-like growth factor receptor, the IGF-1 receptor, then we could get these drugs to actually kill the cells. So this looked like a promising way to proceed in treating these types of tumors. And so before we can take results from the test tube into actual patients, we always have to test these things in animals with tumors to see if it really holds up in a living organism. So what the Connecticut BHI funding has allowed me to do is to conduct animal experiments of these combinations in animals that are carrying human breast tumors that have been implanted in the animal. And so far the results are looking very promising for these combinations. The last part that we're working on just more recently is actually triple drug combinations, combining Herceptin type drugs with anti-estrogen type drugs with the IGF-1 receptor inhibitor drugs in a triple combination. And so far this is being done in the test tube, and the results again look dramatically more synergistic than even the double combinations of drugs. And that's what we've been working on, and we hope that these results will someday soon show us what are the best drug combinations to take into clinical trials in patients with breast cancer to take the next step forward. So some of the major pharmaceutical companies have been interested in developing drugs to treat patients that are drugs that inhibit the IGF-1 receptor. And after my first year or so of research on the Connecticut BHI project with these exciting preliminary results, one of the companies that's developing a drug is Pfizer, and I presented the research that I had done with this funding to the members of the Pfizer development team developing this new drug. And they were interested enough to give me their new drug to work with, and also to give me additional funding at the level of $125,000 for another 1 year to further develop the project using their drug specifically. And that's a great example to show how starting with a small pot of money can generate the idea that can then lead to the next step and larger amounts of funding from other agencies as well.